forgot the acknowledgements. That was bad.
Morning, everyone, and thank you for this wonderful opportunity to allow me to present our work here today. So a little bit about the background. So the information associated microvascular thrombosis occurs in many diseases which can cause organ dysfunction and even mortality. However, our understanding of microvascular thrombosis is very limited, due largely in part to lack of appropriate models. Most conventional in vitro models culture endothelial cells statically on 2D substrate, which are not physiological. Animal models with intra vitro microscopy has difficulties for assessing microvasculature deep inside the tissue. And also, animal endothelium does not exhibit the phenotype same as human endothelium. So to complement these models, we have developed hydrogel-based microvasculature-on-Chip technology with human primary endothelial cells. And it allows to quantify endothelial permeability under physiological flow condition for long term. It also has self-healing endothelial barrier function, and it allows us to study the clot resolution. So here is an image at the end of the permeability assay in which we perfused the flow-tagged albumin into the engineered microvasculature. And you can see here it exhibits an excellent barrier function. We also found that endothelial cells express continuous value coherence, demonstrating the physiological formation of barrier function. We found that the barrier function of the hydrogel microvasculature-on-Chip is disrupted by inflammatory factor TNF-alpha, demonstrated by the increased permeability shown here. But it self-heals over time after the insult is removed. And we also found the permeability return back to the baseline about 4 days later. So when we perfused the whole blood into the engineered microvasculature, we found in the control condition, perfusion of the whole blood didn't cause thrombosis. However, in the infant microvasculature, you can see here, I think that I inserted the wrong video here, but basically when we perfused, so this is the end of the video. So basically the video tells us that when we perfuse whole blood into the infant microvasculature, platelets aggregates forms in minutes, which is accompanied with gradual fibrin formation. Because a lot of studies indicate that platelets aggregation is associated with VWF. So we stent VWF and then find out TNF-alpha induce VWF release from the endothelium. And this VWF multimer are mainly deposits at areas of bifurcation and shear gradient as demonstrated by our computational simulation. This suggests antiplatelet medications that inhibit platelets binding to VWF multimers could be useful in the early stage. We also found out the resolution of inflammatory thrombi is determined by the extent of the inflammation. In the last infant microvasculature, by exposure to 1 nanogram per ml TNF-alpha, you can see that fibrin gradually degrades and neutrophils are also gradually removed. However, in the more severely infant microvasculature, by exposure to 10 nanogram per ml TNF-alpha, fibrin and neutrophil persist post-thrombosis. So this suggests that the control of inflammation could be really helpful for treating thrombosis. And we find out perfusion of TPA induce complete fibrin degradation and neutrophil removal and restoration of the flow, which together facilitate recovery of the endothelial barrier function. This highlights the importance of flow in maintaining endothelial barrier function. And in the end, we found the enoxaparin prophylaxis prevent fiber inflammation, but it does not attenuate microvascular occlusion by neutrophil platelets aggregates. You can see in the end, you can see this aggregation occluded the channel. And also, these aggregates eventually will cause the closure of the occluded microvasculature. So this suggests inhibition of neutrophil platelets aggregation is necessary for treating inflammatory thrombi. So to summarize that, we have developed a microvasculature in vitro model, which allows us to study the step-by-step process of microvascular thrombosis formation and resolution. And we currently study further the underlying mechanisms, and also we try to use this in vitro model to test drugs. And also we are analyzing the embolism formed in our system. With that, I'd like to thank my collaborators and funding source and the whole lab. And thank you very much.